Hi, and thank you for joining. During this short video, we will discuss DNA replication. This video will explain what DNA replication is, as well as when and why DNA replication occurs. We will also explain the concept of lagging strand and compare and contrast the roles and differences between leading and lagging strands in DNA replication. Finally, we will describe the functions of the key enzymes involved in DNA replication. DNA is a double-stranded structure. Each strand of DNA is made up of a sequential array of nucleotides, or nitrogenous bases. There are 4 different nucleotides in DNA: adenine, guanine, thymine, and cytosine. Each nucleotide is accompanied by a deoxyribose sugar and a phosphate. Adenine and guanine are purine bases, while cytosine and thymine are pyrimidines. These nucleotides pair with each other, creating the famous double helix. It is important to remember that adenine only pairs with thymine and that guanine only pairs with cytosine. DNA strands have directionality, and the different ends of a single strand are called the 3' end and the 5' end. The strands of the double helix are anti-parallel, with one being 5' to 3' and the opposite strand being 3' to 5'. Directionality plays a role in DNA synthesis because DNA polymerase can only synthesize DNA by adding nucleotides to the 3' end of a DNA strand. DNA replication is the process whereby an organism's genetic material gets copied. If you recall, cell division occurs to replace old, damaged, or dead cells, and during the process of cell division, all the information in a cell has to be copied. Since the DNA lives inside the cell, it needs to be replicated or copied to make exact copies of itself whenever cell division occurs. DNA replication happens during the synthesis phase of the cell cycle, right before the start of mitosis or meiosis I. Each DNA molecule has many origins of replication sites, all of which initiate the copying of DNA. Why do we need so many sites? DNA molecules contain millions of bases, so having multiple sites reduces the time it takes to copy the entire genome. Think of having to photocopy tons of pages. It's easier if you can split the work between you and some friends using various photocopy machines and then combine the sections once everyone's done, right? That is essentially what happens with DNA replication. The process of DNA replication is said to be semi-conservative. What does that mean? It means that during the copying process, each daughter strand retains one of the parental strands. So you basically separate the parent strands and use each of them as a template for a complementary new strand. In essence, half of the DNA is conserved, or from the original molecule, the pink strand in the image. The other half of the DNA is new, the blue strand, and complementary to the original. There are several enzymes that help prepare the DNA molecule for replication. Let's take a closer look at what they are. DNA helicase unzips the double helix. What do we mean by this? The DNA helicase separates the 2 strands of DNA to allow each individual strand to be copied. Topoisomerases relax the supercoiled DNA by making tiny cuts. For the DNA to fit inside the cell, it needs to be supercoiled. Think of a phone cord or a rubber band that has been twisted so much that it becomes a little ball. If we try to straighten one end of the coil, other areas might become even more tangled. To prevent this, the topoisomerase comes in and makes tiny cuts in strategic places to alleviate the coiling pressure so that the strands can be untangled. The single-strand binding proteins help keep the 2 strands separated from one another. They position themselves along the length of the molecule to prevent it from coiling again. DNA polymerase is perhaps the star enzyme in the process of DNA replication. This enzyme is in charge of building a new DNA strand that are complementary to the parent strands. The enzyme, however, needs a little help in the form of a primer to get the work started. A primer is simply a short string of nucleic acids, specifically RNA, that binds to specific locations in the DNA molecule to provide a starting point for the polymerase. The polymerase then uses that primer to build upon by reading the parent strand and bringing complementary bases to extend the new DNA strand. For example, if the parent strand has an A, the polymerase brings in a T to form the new strand. If the next nucleotide is a C, the polymerase will bring a G to the new strand, etc. An important fact about DNA polymerase is that it proofreads or checks its work to make sure that the number of mistakes is minimized. Another important thing to know is that this enzyme only works in the 5' to 3' direction, which is the reason there is a leading and a lagging strand. Once DNA is unzipped and untangled, and two strands are held apart, a replication fork becomes apparent. A complementary strand for each of the parent strands needs to be made, but if you recall from the previous slide, the polymerase only works in the 5' to 3' direction and creates what is known as the leading strand. So, how does it make the second complementary strand that goes from 3' to 5'? This second strand, which is known as the lagging strand, is built by using multiple primers rather than just one. DNA polymerase then adds pieces of DNA called Okazaki fragments to the space between each of the primers, creating a discontinuous or fragmented DNA strand. As mentioned previously, the leading strand is built as one continuous strand, whereas the lagging strand is built in fragments known as Okazaki fragments. When the leading strand is being built, an enzyme known as RNA primase places a single primer on the 3' end of the parent strand, and the enzyme known as DNA polymerase III starts to bring complementary nucleotides or bases to build a complementary 5' to 3' strand. Once this process is complete, an enzyme known as DNA polymerase I replaces the RNA primer with DNA. The lagging strand is built a little different and has one additional step. First, the RNA primase places multiple primers along the parent strand. Then, the DNA polymerase III adds complementary bases for each of those fragments. Once that process is completed, DNA polymerase I replaces the RNA primer with DNA. As a final step, the DNA ligase enzyme links the Okazaki fragments together to build one continuous strand. DNA replication is the production of two identical daughter DNA molecules from one parent DNA molecule. In order to do this, the parent DNA strands are separated and a replication fork is formed, which allows each strand to serve as a template for new strands. Primers bind to the DNA, and DNA polymerases add new nucleotide sequences in the 5' to 3' direction. This addition of nucleotides happens in a continuous manner in the leading strand and in a fragmented manner in the lagging strand. Once elongation of the DNA strands is complete, the strands are checked for errors and, if necessary, repairs are made.